but it wasn't a bad thing. It just wasn't the one I wanted you to watch. Um, so we're going to do a Nearpod first, and then we're going to watch the one you should have watched that actually did go along with the notes in Canvas. So as soon as I get all these other things launched, I will give you the Nearpod. Just finished watching the tail end of the Jimmy Carter funeral. That was kind of sad. And I know y'all are miles wide close to Plains, Georgia. Anybody been to Plains, Georgia? Anybody actually seen him preach in person? Nobody? I know y'all kind of young. That's a president that was before your time. All right, I'm putting the Nearpod link in the chat. I do need everybody to participate. So you know how Nearpods go. They give you a little chit chat and then they ask you to give a response. So I will be looking for everybody who's logged in to respond. Okay, let me pull this up. All right, so the link is in the chat and everybody should go ahead and log in. So far, I got 3 people logged in and participation in on a Nearpod is recorded. So you do want to make sure that your uh, presence is noted. I got 5 people. And I should have 8. So 3 people are still missing. All right, somebody's got a cat. All right, and again, just housekeeping rules. Everybody should be on camera. Everybody should be participating. When we do assignments this semester, um, you have enough class time to get started on these things. We're not going to deal with late assignments. If they're late and you submit them late, they're going to be marked late because we're not going to have that backlash of people trying to catch up and doing extra work and all that sort of stuff, unless you have an excuse from the principal as to why you should be given special considerations. Okay, so we need you to stay on top of things. All right, I got somebody's still missing. And here again, this comes from people logging in and then taking a walk. Okay, but like I said, your participation is needed on every slide that requires one, so it will be noted as to whether you were here and not participating. All right, so I'm going to go ahead and get started. Like I said, I got 7 out of 8 people logged in, and we're just going to keep on going here. All right, so our objective is to use a model to explain why asexual reproduction results in offspring of identical genetic information, and sexual reproduction results in offspring with genetic variations. That's the objective. All right. First, is this the right one? This is not the right one. I did it again. Yeah, this is not the right one. So I'm going to come out of this one. All right, give me a minute. And I have to get you to the right one. Because we're going to come back to this one. This one, we haven't covered what we need to do on Punnett squares yet. Okay. Um, minus. It should be. This one. Yeah, this one. So let me give you a different code. Sorry about that. There's so many of these things, and they all look alike. All right, so use the second link that I just put in the chat and uh, join that one. Yep, this is the right one. All right, and we lost somebody already. Hopefully she'll come back. All right, so this is the second link in the chat that ends in 4D6Q5. That's the one you need to click on, and please join this Nearpod. I do apologize for submitting the other one wrong. All right, so everybody please join this one. This also tests to see whether you're with me or not with me. All right, I'm waiting another minute for you to log in because the almost immediately they're going to ask you a question. So I need you to be logged in to this Nearpod. Let me see who's missing. Jacoby, I need you logged in, and who else? I got you. Okay, still missing somebody. Kendall, LaCoria. Please log in. Okay, I'm going to get started.
Genetics helps us understand the biological programming behind all life forms. But what exactly is the science of genetics? And what does its future hold?
All right, so go ahead and answer that question. That's your first participation question. All right, so we have talked about meiosis and mitosis, and we've talked about DNA and cell replication. So, based on what you've already covered, what would you say that you already know about genetics? And you're not allowed to say, I don't know, because you know something. Mm, I'm gonna learn to get a clock that times you. So we have, so give you another minute to type in an answer. And as I said yesterday, this is a unit that has a lot of vocabulary, and it is important that you study the vocabulary because you have to recognize what questions are asking you to answer based on the vocabulary words. So be constantly going back and forth, making sure that you understand the vocabulary. All right, we still have people with no answers. We'll work on it. So, all right, so we have one that says some genetics is about how kids get their traits from their parents. They make your DNA and you and stuff. How genes and traits are passed down from one generation to the next. How genes carry information that are passed from one generation to the next. And there are different types of, okay, and not some different types of genetics, but different types of genes. Genetics makes up a person. That's a good answer. I like that. All right. Aaliyah, what you doing? What you doing? Where you at? I see your camera moving, so you must be there. I need you to answer that. All right, we're gonna go on.
Genetics is the study of heredity, the expression of traits and how they are passed from generation to generation. For thousands of years, humans have observed this inheritance of traits and implemented their knowledge to breed and domesticate plants and animals. However, the science behind inheritance was only starting to be understood in the mid 19th century. Around 1865, Austrian monk and botanist Gregor Mendel published the results of his hybridization studies of pea plants. In his findings, he noted the role of factors that influence the expression of traits. These factors later became known as genes.
Okay, so next question. Next question. Who discovered the factors that influence the expression of traits in his hybridization of the study of pea plants? All right. Now, every type of science that we have always has someone who is considered the father of that particular branch of science. So this gentleman, he is considered the father of genetics. Okay. All right, folks, we shouldn't have non-participation. Everybody should be giving an answer. Everybody give an answer. So who is the father of genetics? Got one no answer. There we go. Thank you so much. All right, so yes, Gregor Mendel, he was an Austrian monk, and in the monastery they had a garden, so he did all of his studies there. You do have to remember his name.
Each human has between 20,000 and 25,000 genes. This collection, called a genome, determines a person's traits by influencing factors on a cellular level. Genetic information is stored in every cell's nucleus. Structures called chromosomes carry this information in the form of deoxyribonucleic acid, or DNA.
All right. Genetic information is stored in every cell's what? Again, looking for everybody to participate in a timely fashion. Okay. Come on, folks. Everybody's supposed to be following along, and when an answer is needed, you need to answer. All right. The correct answer is the nucleus. Most of you got that. We still have somebody who's a non-participant, or you're sitting there waiting for us to give the right answer, and then you click on it. All right, the nucleus is the center of a cell. So we talked about the cell structure in the past and what the function of all these organelles are, and that all the chromosomes are located in the nucleus. And think of the chromosomes as being a string of pearls, and each pearl on that string is a gene that codes for a particular trait. So that's what we are looking at. So all of that is in the nucleus.
DNA is a double helix of nucleotides, chemical compounds composed of sugar and phosphate molecules, along with the bases thymine, adenine, guanine, and cytosine. These segments of DNA are what we call genes, and it is within those genes that chemical compounds provide the coding for all information about a person's inherited traits. Human cells contain so much DNA to carry this large amount of information that, if unraveled, the DNA in each cell would be over 6 feet long.
Okay. So easy question. What is deoxyribonucleic acid, or DNA? 1 minute. These answers have to be read thoroughly, so give you extra time to read it and make a selection. And I might add that this particular Nearpod is more of a review than it is going forward with new information. So all of this, we've talked about DNA, we've talked about nucleotides, we've talked about the sequencing and replicating the genetic material. All right, what we got? All right, half of you say it is a double helix of nucleotides chemical compound composed of sugar, phosphate in the nucleus, along with the bases thymine, adenine, and guanine. That is the correct answer. All right, it is not single-stranded. It is double-stranded for sure. Okay.
At the turn of the 21st century, an international effort to decode human DNA was launched. Called the Human Genome Project, it ended up identifying about 99% of the entire human genetic sequence.
A revolution in medical science whose implications far surpass even the discovery of antibiotics.
Okay. True or false? In the turn of the 21st century, international efforts to decode DNA was launched and called the Human Genome Project. One of you actually did your project last semester on this. We were looking at genetic techniques. This is true. Somebody's still not answering.
Discoveries in genetics research have unearthed tremendous opportunities in medicine, such as genetic testing and the manipulation of genes. But with these opportunities come risks and ethical questions. And finding the answers to those questions may be the next stage of our understanding of genetics.
Okay. All right, I'm going to skip this question. Actually, no, I'm not. Okay, she's back, but she missed the whole video. All right, go ahead and answer this question. So we did some things with genes last semester. Write down something that maybe not the video didn't make you curious about, but what are you curious as we go into this topic of genetics and traits? What are you curious about? So everybody go ahead and answer that question. All right, Michaela, I know you just came back, but you can join and just answer this question. So everybody needs to answer. What are you curious about? Genetics is a very curious topic. What are you curious about? Oops. Okay, didn't mean to press the wrong button. All right, so we have, I'm curious about more information, how it works, determine things like eye color and height, and we are definitely going to figure that out. Specific traits, how they happen, reality. You can't say nothing. Gotta be curious about something. All right, so that is the end of that near part. All right, I want you to go to Canvas now, and we're going to take that module step by step. Where are these not? There they are. All right, so again, the overview had all of the vocabulary. So as we go through this particular section today, you're going to see that almost half of that vocabulary list is going to be introduced just in this section on Mendel's law. So I need everybody to turn to that section. And actually, if you would, and this video is very short and I don't even know why it's there, but take a few minutes to read that first paragraph, and I'm going to go through this genetics primer with you.
Genetics primer. Mental and modern genetics terms.
Okay, I'm going to leave it right there. I'm going to stop sharing so I can do attendance, and then I'll be right back. Can you get the link to Canvas or the Nearpod? I'm sorry, if it was the Nearpod, don't worry about it, but just go to Canvas. Another thing I'd like for you all to do sometimes is when you have, because sometimes if I'm reading something within the class, I don't see what you type in the chat. So it is okay to just come off the mic for a minute, unmute and just say, excuse me, can I have a question? And so that you get my attention because I'm not always looking at the chat to see that you have a question. And I don't want it to be so long delayed that it becomes useless. So if you have an immediate question, you need to come off mute and ask that question, please. All right, so read that paragraph, read this paragraph, and I'm going to go through this section with you on the genetics primer as soon as I finish attendance. Okay, and it looks like all right, it looks like I only got one missing person. We have some folks having some technical difficulties. All right, but everybody should be in the genetics module. We have one person absent, so this is not going to take long. Oh, okay. It's participation, Michaela. That there were no real things for you to answer on that one. All right, LaCoria. Okay, I got you in one time now. You having laptop issues? Okay, I'm going. All right, so I am back. All right, and I think I kind of went over this first part with you yesterday as far as selective breeding is concerned. So what normally in nature, things breed on their own. But in the case of Mendel, he was specifically wanting to study specific characteristics. So he selected which organisms he wanted to work with. He was particularly looking for certain traits to show up. And so he did what we call selective breeding. Okay. All right. So let's look at this. And again, there's going to be a lot of vocabulary in here. So please take note.
Genetics. Mendel was an Austrian monk and he lived from 1822 to 1884. His genius was that he noticed differences in peas being grown in the monastery's garden and applied the scientific method to determine the cause. Mendel chose the garden pea Pisum sativum because peas were easy to grow, they had a short generation time, they had a number of contrasting traits that were easy to distinguish, such as short versus tall, or white versus purple flowers. And they also normally self-pollinated, but could be cross-pollinated.
Sound is too low. Okay.
Able to control mating this way by removing the male or the female
Okay.
flower parts and cross-pollinating with a paintbrush. Normally, peas do cross-fertilize or fertilize themselves, but he didn't want this to happen. Mendel didn't really know about inheritance. No one had great ideas about it at that point in time. We knew cells were, but we did not know about organelles, protein synthesis,
Is that better?
or even genes or chromosomes yet. All Mendel had to go by was physical traits and his knowledge of flowers and statistics.
Okay. All right. Hopefully you were following along with that, even though I wasn't there. Okay, so basically what he's talking in this particular section is that he studied pea plants and he studied characteristics or traits such as short plants, tall plants, the color of the flowers, and the texture of the flowers. And he did this by normally, like I said, like they say here, they would self-pollinate, meaning that the male structure of the flower would simply deposit the pollen in the female structure so that the eggs could be fertilized. But he used a paintbrush so that he purposely transferred the pollen to the female structure so that he could get pollination. And he did that because he wanted specific plants to pollinate certain plants. What happens when they talk about birds and the bees, what happens in nature is that the pollen sticks to the fur of the bird or the bee, and when it moves to another flower, then some of that pollen falls off, and this is what we call cross-pollination in nature, but he did it on purpose.
Mendel chose easy to recognize, true breeding varieties for his experiments, some of which are pictured here. True breeding, or purebred, means that when the plants were bred, offspring maintained that characteristic over time. Therefore, the offspring showed traits identical to their parents and identical to each other.
Okay, so when we look, the traits that he's studying are the seed, type of seed that the pea plant has. So there's different forms. They can be gray and round, or they can be white and wrinkled. They can be cotyledons. Cotyledon means is the type of seed, and we'll talk about plants later, but the seed splits in half. If you've ever had green peas, you know that if you kind of mash it a little bit, it kind of has two sections to it. So what do the cotyledons look like? So are they yellow? Or are they green? What does the flower look like? Is it white or is it purple? Um, the pea pods that the peas actually grow in, are they full or are they constricted, meaning kind of wrinkled? Or are they yellow or are they green? Okay, notice the cotyledon and the pod color coincide, so they're going to be the same. The stem, the place of the leaves on the stem, is the arrangement of when they say axil, are they arranged straight up and down, or are they oft kind of going in a pattern, a circular pattern kind of thing? Are they terminal with the flower? And then the stem, is it long or short? Okay, so looking at the way this chart is, you can see that the round ones are going to make yellow cotyledons. They come from white flowers. They're going to form a green or yellow pod, and they're going to either come from a tall or short plant. So those are the characteristics that we're looking at.
Mendel examined genetics in peas over generations and kept detailed records. For instance, you can see in this diagram that the parent or the pea generation are pure breeding purple and white flowered plants. They were crossbred with each other. This was done carefully by use of a paintbrush to transfer the pollen from the male parts of one flower to the female parts of the other flower. The offspring that result from the pea generation cross are called the first filial or F1 generation. Then, if members of the F1 generation are bred with each other, their offspring were called the F2 generation or the second filial generation.
Okay, so this is our first set of intense vocabulary. So we're going to be looking at predicting the possibilities of the kind of offspring that can be produced by crossing one type of organism with another type of organism. In this case, he is looking at a white flowered pea plant with a purple flowered pea plant. And the little X in the middle means that we're crossing them or we're breeding them. And so you need to be familiar with this because you're going to have to write your I don't know how to put this, your expressions of traits just like this. So you're going to have to show the parents, who are you crossing to begin with, and then what their offspring are going to look like. So we call the first ones the parent generation or the P generation or the filial generation. And then their offsprings are the first filial generation. Okay, so that's what F1 stands for, first filial generation. They are the offsprings that will result from crossing these two. Now, when we did meiosis and mitosis, we learned that meiosis creates 4 cells. So that means there's going to be 4 possibilities of what we can get from crossing one parent with another. So when we actually study these or map this, we're going to look at these as what are the 4 possibilities. They're only showing you one right here, but we're going to actually draw it as 4 different possibilities, even in the F1 generation. And then we'll take 1 or 2 of those offspring from that one and cross them to get an F2 generation, which will be slightly different than our F1.
Let's take a closer look at this same experiment. In the P1 generation, Mendel uses his paintbrush to control pollination. The true breeding white flowered plants are bred with the true breeding purple flowered plants. The F1 generation that results from Mendel's cross all have purple flowers. Then Mendel allowed the F1 generation plants to do something they would normally do, self-pollinate. This results in the F2 generation at the bottom of the picture. In the F2 generation, the white trait reappears. This was interesting because it meant that the white trait was still present in the F1 generation, but was just masked or hidden somehow. Mendel called these traits that were passed down over generations inheritance factors. We now call these alleles. It's important to remember that Mendel had a very strong mathematical background. He traced the inheritance of each of the individual traits in peas and kept very careful records of the numbers. He used his understanding of statistics and probability to make conclusions about how the traits in peas were inherited.
Okay, so we have a vocabulary word, allele, and I don't know if you remember that word allele from when we were talking about meiosis and mitosis. The chromosomes, they lined up with like chromosomes. So you have one chromosome that might have the gene for tallness on it. And you get one chromosome from mom and one chromosome from dad, or from the male structure and the female structure. And they have to line up. So the gene for eye color might be on a different chromosome. The gene for curly hair might be on a different chromosome. So the ones that have the same characteristics pair up. Those are your alleles, so the chromosomes that are alike, that have like information. When we say like information, again, type of hair, type of color of eyes, tall or short, those are going to line up together. Now, this one may say tall, this one may say short, this one may say blue eyes, this one may say brown eyes, but the ones that have that characteristic, they're going to pair up. Okay, so you have to remember alleles. All right. The other thing that they've talked about is dominance and recessive. There are certain traits that, so we're getting one from mom, one from dad. One of them might be a stronger characteristic than the other, and that's the one we call dominant. And the other one is there, but it doesn't show up. So that one is the one we call recessive. But they're still going to pair up. And she used the term mass. One trait masks the other. That's like a tall person and a short person standing one in front of the other. The short person is there, but the tall person is the only one we see. So the tall person becomes the dominant characteristic and the short person becomes the recessive characteristic. Again, they are there, but we don't see they're not expressed.
Mendel published a paper in 1865 with these three conclusions to his research: the principle of dominance and recessiveness, the principle of segregation, and the principle of independent assortment. The principle of dominance and recessiveness states that one factor in any trait's pair may mask the effect of another. We examined this on the last diagram, when the traits from one generation reappear in later generations. The principle of segregation states that the two factors for a trait separate during the formation of eggs and sperm. Remember that this happens during meiosis when the parent's homologs separate to make gametes. The principle of independent assortment stated that the factors for different traits are distributed to the reproductive cells independently of each other. This means that the offspring inherit a random mixture of the different traits that are present in their parents. At the time of Mendel's paper, chromosomes and the process of meiosis were unknown. Mendel based all of his conclusions on mathematics rather than an understanding of genes. Mendel's work was considered obscure and unimportant until the 1900s, when it was rediscovered and made important connections with the chromosome theory.
Okay, so these are three basic principles, and you are going to have to be able to identify these, but we'll do these in sections. So we'll do an activity that basically illustrates dominance and recessive. We'll do an activity that illustrates segregation, and we'll do one that illustrates independent assortment.
Modern genetics primer. Since the days of Mendel, a number of discoveries have been made that help us better explain what Mendel was witnessing in his experiments. Namely, we now know what DNA is, we know how meiosis works, and we can put these pieces together to come up with some vocabulary to describe the genetics behind Mendel's experiments. A phenotype is the external appearance of an organism that is inherited. It refers to the physical appearance of the individual that is coded for in its genes. Phenotypes can be colors, as in Mendel's pea flowers, or a variety of possible patterns, as you can see in these bivalve shells, or even physiological effects, such as lactose intolerance. Lactose intolerance is caused by the lack of a functional enzyme that's supposed to be digesting the lactose sugar in milk. Remember that the outward appearance of traits, or the phenotype, is always coded for in the DNA.
Okay, so the vocabulary word that you have to know. And it's important that you understand what this one is, because then there's the next word that she's going to discuss, and you have to know the difference between the two. So when we actually start mapping, you're going to get two questions, and it's going to ask you, as a result of what you've mapped, what is the phenotype? And that means what based on the information you've given, what does this organism look like? You're going to have an activity where you're going to have to create something and discuss its phenotype and genotype.
We now know that Mendel's factors are actually the genes that are inherited from mom and dad. We can also call these alleles. So for each trait, such as flower color or even tongue rolling, each offspring receives 2 copies, one from each parent. The combination of these alleles makes up the genotype of an individual, which we will talk about next.
All right, so again, she's showing that the chromosomes match up. You see that letter P, that represents the gene for flower. Again, think of genes as individual units on a chromosome, and each unit is coding for a different trait. In this case, these letter P's stand for the flower color. So and a chromosome with a particular trait on it is an allele. So again, vocabulary, allele. Know what an allele is.
How can we represent alleles? Well, there are 2 types of alleles. The first is dominant, and those mask the expression of another trait. We will use capital letters in this course to denote a dominant allele. Recessive alleles are masked if a dominant trait is present. Remember that the white flower was masked by the purple trait. So the white trait is recessive, and we'll use small letters to denote recessive traits. If you have at least one copy of a gene that works, or a dominant trait, one that produces purple flowers or purple pigment, you can still produce the dominant trait. So you can see underneath the purple flower picture, we can make a combination of alleles that have at least 1, but possibly 2 capital letters. These would both show the dominant purple trait. For a white flower, the only allele combination would be 2 small letters, or 2 recessive alleles to show the white phenotype, or outward appearance.
Oh.
The letter that we use to represent the gene doesn't have to be P. In fact, it's arbitrary. We could have used A if we wanted to. A couple other things that you need to know. Dominant genes don't necessarily suppress recessive genes. The recessive gene is usually an absence or a deficiency of some protein. Also, dominant genes aren't necessarily more common. Some of them actually cause disease, such as Huntington's disease.
Okay. One thing she just said that we're not going to follow is that the letter that we use doesn't matter. Typically, as a rule, they use the capital letter of the dominant trait. So in this case, purple is dominant. And so they use the capital letter P for purple so that you can, okay, looking at people coming and going. The capital P is for the dominant trait. And we don't just write a small w for the recessive trait. We use the lowercase p, to represent the recessive trait. And again, both are present, but we're going to have a capital, and notice there's 2 ways that we can express the dominant trait. We can have capital P, or a capital P and a lowercase p. And that both of those will express as purple. If you do the lowercase, that's the only way that you will see a white flower. So in order for expression to occur in a recessive characteristic, you have to have both recessive genes. Okay, everybody follow before I go to the next one? So again, vocabulary. You want to know what is the difference between dominant and the difference between recessive. Okay. All right, we good? All right, so we've got alleles, we've got dominance, we got recessive, we've got phenotype, and now we're looking at genotypes. We're looking at what genes are present. So the letter P's are expression of the genes. Saying that it's purple is expression of its phenotype?
The genotype is the genetic makeup of an organism. It refers to the letters or the alleles that an individual received at fertilization, one letter or one allele from each of its parents. The purple flower has two possible genotypes. It could have two capital letters to make up one genotype, or a capital and a small letter. The white flower has only one possible genotype. It would be made up of two small letters.
Okay. So again, the term genotype only describes what genes are present. And it's important that you remember this, because when we do it as an activity, the question will specifically ask you, what is the genotype? It will specifically ask you, based on what you wrote, what is the phenotype? So you need to know that when I ask you what is the genotype, you're telling me what genes are present.
There are three kinds of genotypes. The first is homozygous dominant. In this genotype, there are two dominant alleles for a trait, such as capital P to represent purple flowers. The homozygous recessive genotype would be a genotype that's made up of two small letters, or two recessive alleles for a trait, like you would see in white flowers. Finally, there's a heterozygous genotype. In this genotype, you would see one of each type of allele for a particular trait. So you would see a capital and a small letter in combination.
Okay, so we've got three vocabulary words that you have to, and again, you'll be asked to express them, the genotype, in this fashion. So if I have two capital P's, homo means same. So if they're both capital letters, then they are homozygous dominant. Dominant because they're capital letters. If they are skipping down to the bottom, let me not skip to the bottom, let me do the homozygous recessive, again, meaning the same. But in this case, recessive means we've got two of the lowercase letters. Okay, so they are the same trait and they are the same size. Heterozygous means that the letters are different. We have one dominant gene and one recessive gene, but it's still going to be the purple. So we're going to say heterozygous purple, or we can say homozygous purple. If I say homozygous purple, that's telling me that both genes are the dominant characteristic. If I say heterozygous purple, that's telling me that I have two different genes. If I say homozygous white, that's telling me I have both recessive genes. Okay, so the vocabulary matters. So please, please, please learn the vocabulary.
Finally, let's apply the terminology we now understand to Mendel's work with pea plants. You can see in the purple box what we do know. White flowers are recessive to purple. Therefore, their genotype is small p, small p. Purple flowers are dominant and must contain at least one dominant or capital letter. Their genotype would be big P, or big P, little p.
Okay, so you see in that purple box, they have two things. They have white flowers, and so the white flowers would be the phenotype, and the fact that you have two little p's, that would be the genotype. Okay, the second one, the purple flowers, you have two ways of saying it. Saying purple flowers is its phenotype, expressing it as capital P, or capital P, lowercase p is its genotype. Okay, all right, so we're going to look at a little more as to how you do that.
Recall from earlier that I told you in the pea generation that Mendel used purebred plants. This means that the white flowers must have been homozygous recessive, or small p, small p. The purple flowers, then, must have been homozygous dominant, or big P, big P.
Okay, so again, what the letters tell you gives you what you would call it. So we know that if it has if it's a white flower, we know it's homozygous recessive. If it's a purple flower and it produces only purple flowers, then we know that the parent flower had to be homozygous dominant, because during meiosis, remember the alleles split. So one of the alleles is going to one cell, the other allele is going to the other one. So in this case, one capital P is going one way, the other capital P is going the other way. And it's the only thing it can give is a capital P. So in the first generation, the only thing it's going to receive from that parent is a capital P. The only thing it can receive from the other parent is a small p when meiosis takes place. Okay. Now, when the next generation happens, these parents now have two possibilities. So again, we're going to be looking at possibilities. What are the possibilities that the capital P went to one cell? What is the possibility that you're going to be uniting with a lowercase p? And in which case you get this next generation down into the bottom where they're not all purple. One of them actually produced a white flower. So we're going to look at how that can happen.
Each of the parents in the P generation donated one of their letters to the F1 generation. This resulted in a heterozygous dominant genotype, or a genotype in which you had a big and a small letter. In the F2 generation, the white phenotype reappears. Therefore, it must have the small p genotype, or homozygous recessive. The other purple flowers will either be homozygous dominant or heterozygous. This means that they'll either be big P or big P little p. In either case, at least one big letter must be present in order to show that dominant purple characteristic.
Okay, so again, in the second generation, the possibility of having purple flowers is only what we call 75%. And we're going to be doing this in percentages. So in our first generation, 100% of the offspring are going to be heterozygous dominant, because that's the only possibility that can occur, because each parent can only give one of each characteristic. So that's what we're going to have in the first generation. But in the second generation, if we take these two members of the first generation and pollinate those, the possibility that one parent can give a capital letter and one parent can give a small letter. So the possibility of these letters recombining is different. So we can have a capital P purple flower. We can have a capital P small p purple flower, or we can have a small p white flower based in the second generation. So when we map this out, that's going to be our next thing to do, is to learn how to map this out and predict what the results will be, kind of creating a visual. All right, so what I want you to do with the 3 minutes that's left is to work on this challenge. Now, you may not finish, but we'll finish it later on. And I'm going to base a quiz on the answers to this challenge. So I need you to really, really go through and work on that. And you can check your answers on this, so that when you see it on a quiz, you'll have already figured out what the right answers are. Okay. So like I said, there's only a few minutes left, so you probably only have enough time to basically get it started. But do get it started. At least there's 2 pages to this, 3 pages to this, so at least get the first page done. Yes, I can. Okay, I took care of that. All right, so I don't know if you noticed, Miss Martin kind of popped in for a visit, and hopefully she saw all of you on camera. I know one person wasn't, but kind of engaged in active learning, and they do that from time to time. I also, again, want to remind you that this is end of course test subject, so you want to make sure that you are saving your notes, that you are trying to stay on top of your vocabulary. Later on, I'm going to give you a specific review sheet so that you know exactly what areas you need to be prepping for the end of course test. The the state end of course test, not the my personal end of course test. You have to do both. Okay, and again, when I look at Go Guardian, I need to see everybody doing what they're supposed to do. And I see two people who have not even close to Go Guardian. I mean to Canvas, and everybody should be in Canvas doing the practice drill. All right. So yes, all the vocabularies on the first page of the module. So you might need to go back and forth and review that. So I won't see you until next week, but I will use these as the questions for a quiz. So make sure that that's what you're looking for. Michaela, you look like you're looking at the wrong page. It should be the second page of the genetics module.